everyone, I'm Jenna,
And my name's Steve.
and welcome to another Field Trip Friday. Where are we going today, Steve?
Well, this week we're going to go to a couple places. We're going to UNC to visit the Mitchell Lab, and these folks, they do research on fungal infections in plants, specifically grasses, and we're going to look at a particular grass called fescue. And then we're also going to be going to their field site where they actually plant these plants and where they look at these kind of fungal infections in a natural environment in Duke Forest. And so they do their field research at Duke Forest.
Very cool. I'm excited to see fescue again in one of our Field Trip Fridays.
Absolutely. We keep running into fescue. It's an important grass.
Yeah, let's go take a look.
Let's do it.
So, hey, Brooklyn, Elizabeth, thanks for having me. Who are you and what do you do?
So we are in the Mitchell Lab right now at the University of North Carolina. I'm Elizabeth. I'm a second-year PhD student here.
Cool.
I'm Brooklyn. I am the lab manager for Dr. Mitchell. Basically what I do is kind of orchestrate a lot of the work and things that go on between the graduate students. I consider myself boots on the ground. So
Brooklyn knows all. Brooklyn at this point has been here.
I have been here
for the longest
longer
time
than anyone because I like it. But essentially, boots on the ground is just how I describe my work. The graduate students come up with the research and what they want to do, and I help make it happen in the lab by giving them the supplies they need, the tools, and I help make it happen in the field.
That's awesome. That's a cool responsibility, right? You're connecting these graduate students and their research goals with the actual logistical feasibility of that, right? Especially with a project like what you told me about, where there's field work, lab work, and there's a lot to
There's a lot of moving parts, and there's a lot of experiment. So we have two graduate students and a postdoc. They can each have their own experiment going, and so I'm kind of a liaison between members of the lab.
Yeah, so we're all using different protocols at different times, and Brooklyn really knows everyone's protocol.
So, Brooklyn, what are these plates with looks like blades of grass on them?
Yeah, so they are blades of grass
Cool.
on them. But the blades of grass have what look like spots, different shapes, different colored looking spots. This is what is known as a disease symptom when it's on a blade of grass. So our lab studies the disease ecology of plants. So we look at diseases infecting plants and how they interact with other diseases or the plants as a whole, and how we can manage.
Yeah, that's awesome. I think Elizabeth had mentioned before, you don't have to think about plants getting sick, but there are diseases, including fungal infections, that affect plants, and that's what you're studying.
Exactly, and you can see the disease symptoms on the leaves, just like we could see if a person were sick.
Right.
So chickenpox is a really popular example. You can see someone is sick with symptoms, and this is the case with what we study in the lab. We use tall fescue, which is a grass you can find in your backyard if you were to go outside. And if you were to go outside during the summer, you would likely see these very same things.
Cool. So, Elizabeth, we have learned a little bit about the fungal infections that y'all study in these plants. What are you specifically looking at today? What are we doing in the lab today?
Yeah, so today we are checking to see if the protocol, which is the method that we use for kind of inoculating or making those plants sick, works. So we have this fungus, we've already grown it, so we're going to put it through a couple of steps to get it ready, and then we're going to apply it to plants and see if they develop these visible lesions, and then we'll know for sure that we've successfully infected them.
Right, so you'll know that you're able to produce this kind of experimental condition, which is an infected plant.
Yes, so then we can do that in the lab, which essentially mimics what is naturally happening outside. And so we're able to test all sorts of variables then.
Right, so these plants would normally just, the fungus would infect them in outside in the wild, but for your experimental setup, you need to be able to make sure that the plants you're experimenting with have this fungal infection. And so to do that, you need to test your method of infecting the plant.
Yes, because sometimes we want one to infect and not the other, so we need to be able to put something exactly where we want it. And what we're doing today is really fundamental for our research, because a lot of our experiments we manipulate, we control which fungus is where, and we use that to test these relationships we're exploring. So if our protocols don't work, we can't use our we can't do the rest of our experiments.
Right. You wouldn't be able to trust whether that plant has the symptoms that you're seeing because of the infection that you planted there, or if not. Yeah.
Yeah.
Right. Like if you have a negative result, you wouldn't know whether that was because it just didn't get infected because your inoculation protocol didn't work, or because of some other condition, which is what you're testing, right? Cool. That makes sense.
So the first step in creating an inoculum is to grow the fungus that we need. So we have part of the fungus growing in what is called an incubator. So it's just a big fancy box that keeps a specific temperature it's warm and nice. There's light that can be provided, and we grow the fungi that we are going to use.
So the next step in our protocol is we take those fungus. So this is one of the fungal samples pulled out of the incubator. And what we would do is we would take off the top and flood it. So put water on the top, and we use this to really scrape the plates, really disturb all that fungus, and that makes sure that we get all of the spores out of the fungus and into the water solution. Which after they've been scraped, they end up looking like this. So that's been scraped and all the water has been sucked back up. And we use that water, which is the beginning of our inoculum. And so we can put it in here, and this is our inoculum solution.
Cool. So we just scraped all the fungal spores into the water solution. What are you doing over here at this microscope?
So the final step in creating our inoculum is just making sure it has the spores. Like we said before, we cannot see the spores with our naked eye. So we do want to confirm that they are there. So we just take some of that water solution we just made, put it underneath a slide under the microscope, and we confirm that we are seeing spores
Cool.
from the fungus.
That's awesome. Cool.
That's our plant.
So the
That's awesome.
plants, once they've been inoculated and they're in their little dew chambers, is what we call them, because it kind of accumulates moisture like dew droplets. It's very similar to what we would see out in nature. They sit in these for about 3 days, and after that point, we can see symptoms. So we'll check them probably daily on days 3, 4, and 5, and we'll begin looking for those spots that we showed you earlier.
Right. And so then once you have infected plants, once you've kind of confirmed you have symptomatic plants, that's when we're going to take them to the field site?
Yeah.
Cool. And so then you can put the symptomatic plants in various conditions to see how those conditions affect infected plants.
Yes, and outside in the field, they're exposed to all the other crazy things going on in nature. So we can look for changes. Maybe these plants become infected with one fungus, but the plants that we didn't infect, they don't become infected with that. So we can see there's a relationship with what we infected in the lab and what we see in the field.
Hey y'all!
Hi.
So we are here at the Duke Campus Farm, right? Is that what we would call this spot?
We would call this a section of Duke Forest.
Okay.
Duke Campus Farm is our neighbor.
Okay, got it.
But we are a separate individual.
I see. So we're here in the Duke Forest.
Yes.
And you have brought some of your inoculated plants. And so this is a site where you do lots of different experiments, right? And we can see behind us that there are all these different plots and kiddie pools and stuff like that. But we're right here in this little area. Can you tell us a little bit about what happens in this area?
Yeah, sure. So this is what we call an outplant experiment. Within the plot, there are a number of holes that we've dug.
Okay.
And so we're able to take our plants, and we have these individual plants, and we can just put the whole plant in the hole. And so that means that we can keep track of each plant over the course of the experiment, because it's all contained within the plants.
Cool.
And the plants themselves are then exposed to all of the environmental variation of being outside. We haven't redug.
Oh, that one was good.
Yeah, look at that. Perfect.
So we have data sheets where we keep track of where all these plants are and what diseases they have, like on week 1. So then next week when we come out, we'll say, okay, last week this plant had only one disease and there were 3 spots of it. There were 3 lesions. This time there's a new disease and there's 2 spots of it. So we'll do that for each plant that's out here. And sometimes that's 100, 120 plants.
Wow.
Wow.
So each plant has an ID and each hole has an ID number, so we know exactly
Where
they've all
each
been.
plant is, and across time. You
Yeah,
can,
yeah,
you can
across
see
time.
where the plants have moved
And
through.
time is a big factor because we're dealing with disease epidemics. So time is really important. As we can see, winter, nothing's happening.
Right.
So we need to be able to
And
know
I think
where they are.
also it's important to mention that fungus, spreads in a really interesting way. Do you want to talk about how fungus is able to
Sure.
move?
So it can actually be pretty diverse. We probably all know what spores are, fungal spores. So that is a really common way for
The like
fungus to spread.
if you have like a mushroom at like a portobello or something, all that
Yeah.
black
And,
stuff
if
like
you-
in their gills.
Yeah, or if you, sometimes it looks a lot like pollen. So if you touch a flower and you see like all this yellow stuff, fungi can reproduce in a similar way. It's just called spores. They're different. So some of them, when the wind blows, they blow spores across all the leaves. Some of them, the other 2 we use, spread via raindrops. So it rains, the rain is pounding, the plant's splashing, and then that actually splashes the spores from one leaf to another.
Whoa.
That's so cool.
Yeah, that's so wild.
And then some of it is just naturally going on in the soil. So some of them just, even the good fungus, they'll just kind of naturally spread through the soil, through the roots and mycelia. Which is why the outplant stuff it's important that they're kind of blending in and touching their natural neighbors here, so that we know they're getting all that good
Yeah.
stuff.
Like Jim.
So I'm curious though that these have like a wire fence barrier plus some
These
pool noodles and
they're
stuff
pool
like
noodles.
that. So why what's making this plot special or different from all these other areas?
So this is another everyday solution, like simple solution to what seems like a really specific problem. But when we first did this experiment, we were coming out here surveying our plants, and then we were realizing our plants were not here. There were like little chomp marks and they were missing. So we realized doing these specific outplant, these plants were really vulnerable to being eaten by field mice and voles. So we installed a fencing that has a small enough size where the rodents can't squeeze through it. Unlike the rest of the plots, they're open to any traffic.
Right.
And then the pool noodles, they help us from getting kind of like nicked by the fencing, but we also felt like it intimidated the mice because it was like bright and squeaky and colorful. So it's kind of twofold. And it also just looks really nice.
Does it work?
Yes.
Okay.
Once we installed this, no more plants, like no more victims. to
Wow.
No more mice victims. So we were coming out and our plants were still here the next week, which was really exciting.
So we're gonna compare 2 things that you have in your hands. So can you tell us a little about there?
Yeah, so this right here is the plate that we showed you guys in the lab. So these are the infection. And so last time when we sprayed all the plants, what we're hoping to get on our leaves here is something that looks kind of like this. So we have a leaf right here.
Oh wow, yeah, I'm seeing it.
Yeah.
So you can see that infection. Yep, so that
Yes.
means that the inoculations that we did worked, and so this plant is now infected.
So when you sprayed all that kind of fungal solution over the
Yep.
plant, lo and behold,
Exactly. So these are the kind of things that when we come out to the field, this is what we're looking for on all of our
Yes.
plants.
And we've been trained to be able to know what they look like. So there are a bunch of different things out here, but we train to know how to identify them by looking at them, so we know these are the same thing.
That is so cool. And we're not just out somewhere in the wilderness, right? We're literally in someone's backyard.
Yeah.
Yep.
kind of anyways, like your neighbor's backyard anyways. Yeah.
We don't walk over there, but this could have very well been their backyard.
Yeah.
So we're answering a lot of big questions that can really be used large scale to help people all over the country in different environments, but we are using stuff right here in our own backyards to answer those big questions.
Yeah.
Cool. Well, thank you again so much for sharing your work with us.
Yeah, Brooklyn, Elizabeth, thank you so much.
Thank you.
Yeah, thank you.
Stay tuned for our live Q&A.
Yep, we'll get to ask a bunch of cool questions of our friends right here. See you there.